We are now going to go ahead and move on to transferases. Okay. In theory, transferases are kind of simple. Is this thing still on? Yeah. Okay, so in theory, transferases are kind of simple. The idea is you are taking a functional group or an atom from one molecule and moving it to another molecule. Okay, and just like with our oxidoreductases, we can break these down into subcategories. So we're thinking about moving a carbon. We're thinking about moving aldehyde or ketone functional groups, acyl functional groups, sugar groups, nitrogen groups, phosphorus groups. Right? Anytime we move a phosphate from ATP to something else, that is characterized as 2.7 because that's moving phosphorus, all kinases, right? Anytime ATP is involved for phosphate transfer, those are categorized as 2.7. Okay, I kind of show in some in red and green here some relevant ones that we're going to look at today. We're going to see a couple examples, at least cofactors that are capable of moving around carbon groups. Okay, there are two really common cofactors for this 2.1 class. We're going to look at those. So acyl transfer, we're not going to look at it today, but next week when we start thinking about translation, this acyl transfer is really, really important because it's one of the very first steps in translation. It's how we load amino acids onto our tRNA molecules. Okay, so this class is incredibly important for translation. We're going to look at nitrogen transfer for these enzymes transaminases, and I'm confident we'll run out of time. So we won't be able to see all of it probably until the beginning of next week, but you'll have all of it available and you can work ahead if you want to. This is really, really critical for the synthesis of every single amino acid and the breakdown of every single amino acid. Okay, and then all kinases are characterized by enzyme class 2.7. All right, so here are some common cofactors. Okay, we mentioned phosphate transfer. That was the class 2.7. All of them use ATP, and the chemistry for phosphate transfer off ATP should not be surprising to you. It is standard, straightforward carbonyl chemistry. Find your nucleophile. The nucleophile is whatever ends up making the bond with phosphate. Your electrophile will always be one of these phosphorus atoms, right? These phosphorus atoms are incredibly electron deficient. They are surrounded by oxygens, and those oxygens are just robbing the electrons. So each of these phosphoruses are incredibly, incredibly electron deficient. They're great electrophiles, okay? The only way of predicting whether we transfer one phosphate or two phosphates or theoretically three phosphates is the positioning of this cofactor in the active site, right? So if the nucleophile is oriented towards our gamma phosphate and the only thing it can attack is this phosphorus atom, then we'll transfer one phosphate. If it is oriented towards the beta phosphate, then we'll transfer two phosphates. Right? And that's really it. Like that is the only predictor of how many phosphates gets transferred. In all cases, it's about the same energy. Okay? Phosphate transfer boils down to ATP typically, sometimes GTP, but usually it's ATP as a phosphate source. Carbon transfer, there's two really common ones. One is this molecule S-adenosylmethionine, and in your activity, you'll look at how this is made. It's really straightforward. The idea is you have an AMP or an ADP or ATP, whatever, but the idea is that in that adenosine, we replace all of the phosphates, including the oxygen on the 5' carbon, with methionine, right? So here is methionine. I somehow left out the carboxylic acid, but so this is methionine, and what you'll notice is the 5' carbon of ribose in our adenosine is covalently bonded to that sulfur. This is the sulfur in the side chain. That's that thioether side chain. And because this sulfur has a positive charge, it's quite reactive. This methyl group is easily donated. It's easily transferred from one place to another. So S-adenosylmethionine, called SAM, is a really good methyl transfer cofactor. The other one is folic acid. Right? So folic acid is quite important for the synthesis of DNA and RNA, for nucleic acid biosynthesis. It's really good at transferring carbon in different oxidation states. It can transfer carbon as a methyl, a formal, or a methylene, right? Which is it boils down to different oxidation states of that carbon. When it's just a methyl group, that methyl is simply attached to carbon 5. If it's a formal group, formate is, right, a carbonyl and a hydrogen. So it is a carbonyl and a hydrogen attached to nitrogen number 10, this position here. If it's methylene, it's a CH2 branching these two, creating another 6-membered ring. Okay, so depending upon what the oxidation state of carbon is present at that position between those two nitrogens will dictate what oxidation state of carbon is transferred using this as a cofactor. Another common one that you've probably heard of is coenzyme A, right? We hear about acetyl-CoA all the time. Acetyl-CoA is absolutely a central cofactor in metabolism. It boils down to an ADP, right? This is ADP right here, coupled to this molecule called pantothenic acid. And what really matters is at the end of it, we have a reactive thiol. That reactive thiol can be coupled to an acetyl group, right? CH3, C double bond O, and then that's attached to that sulfur, okay? That's how we move around 2 carbon units. And then the other one here I want to point out is for amine transfer. And this one is going to become exceptionally important towards the end of the semester when we're talking about amino acid biosynthesis. Because every single amino acid, all of them, boil down, get their nitrogen through an enzyme that uses this cofactor. They're called transaminases. They take an amine from glutamic acid and give it to what's called an alpha keto acid, creating the amino acid, all of them. And they all use this cofactor, pyridoxal phosphate. Okay? So, spend another few minutes working through questions 6 and 7 in your activity, which focus in on our methyl, the cofactors that are good at transferring methyl groups or carbon-containing groups.